This research that we're funding with the University of Missouri and the Broad Institute at MIT and Harvard is actually monies from Purina, and I'm going to have Connie Wagner explain exactly how we're funding this research. Nope.
Just a little louder,
Okay.
please.
So can you hear me now?
Yes.
Just a quick way of how you, because you're actually supporting this. You're the one that's donating the money when you buy food and you put in your receipts. You keep all your points, but 10% of your total goes to George Shepherd's. And if you asked for that, you can opt out of that, but of course, who would want to do that? So 10% of the total in addition to what you keep. Yes, ma'am. Sorry, I'm terrible. So 10% of the total goes to Shepherd's. Half of that goes to the Canine Health Foundation, and half of it goes to Breed Club for the betterment of the breed. So you are actually funding when you send in your receipts. If you're a Pro Club member, that's how they're getting all of this funding. So, you know, you can all pat yourselves on the back because you're the ones doing this funding. And if you have any questions, I'll be at the booth and I'll help anybody to the best of my knowledge.
Yes.
Just wanted to say that Purina has the Purina Weight Circle funds. We get two different kinds of dollars from Purina. We get about $15,000 from Purina during our national time, and that's supposed to be used for our national. But we also have the Purina Weight Circles that when you send in your weight circles and or do whatever it takes to get that donation in, they send you special value certificates so that you can buy Purina at a less discount and the other things that they do. But one of the things that they do based on those weight circles that all of us send in is they give us the Purina Weight Circle money, and it is split between, or doubled actually, as the case may be. We get a certain amount and it goes into our trust, the German Shepherd Dog Club of America Charitable Trust, which is a 501c3, and that money is to be spent on education, it's to be spent on rescue, and it's to be spent on health. An equal amount of money goes to the AKC, and the AKC has been saving
Canine Health
that money
Foundation.
for us for years because Purina has been donating to the AKC for years for us in trust with the AKC, and their fiduciary duty is to keep that money for education, rescue, and health. And this is the first time that we have used any of that money for a large project like this, which is a very large project. So that's where Purina has helped us and partnered. They are really, really and truly our partner and have been for many, many, many years. I don't know if people really appreciate how much they do for us and how much we're able to do because of their donation and their largesse toward us, and we really appreciate it.
I would just like to say that it's not weight circles, it's receipts now. We don't do weight circles anymore. So don't save the weight circles on your bag, but redeem, upload your receipts through your Pro Club, and your points will be in your account in an hour.
Thank you.
Thank you.
Now I'd like to introduce Liz Hansen. She is one of the members of our team for the DM research project, and she can explain what we're hoping to do, what they're doing, how to participate, what type of dogs they are looking for right now.
We need it much louder.
I'm generally loud anyway, so hopefully I'll be loud enough. Okay, so as she said, I'm part of the team at the University of Missouri. I work for Dr. Gary Johnson in the Canine Genetics Lab, and I've been there about 22 years. So what we're going to talk about tonight, we're going to kind of cover quite a few areas of this, I hope. We're going to do a little review of canine degenerative myelopathy, which too many of you are way too familiar with anyway, but we'll go over some of the scientific ends of it. Some of the genetic discovery and testing that we do, some recent clinical and retrospective research, and then talk about the current grant that we're working with that the club here has funded. So, canine degenerative myelopathy was first described in German Shepherds in 1973, and so a lot of vets have learned that DM is a disease of German Shepherds. It's a later age onset disease, generally 9 to 14 years of age, and progressive hindlimb weakness. Most dogs, once they start showing some clinical signs, it's about 1 year until they're basically non-functional. But it can be anywhere from maybe 6 months up to 3 years, and somewhat dependent on what the owner can maintain. I mean, if it's a little bitty dog, you can carry it around, they tend to live longer. If you've got 100 pounds of dog, it's really hard when they can't stand up. So it's harder to maintain them longer. The true absolute diagnosis for DM is to look at the histopathology. Look at a piece of the spinal cord after the dog has been euthanized, and that's the gold standard for a diagnosis. And there is no effective treatment at this time, unfortunately. So what's going on here in the dogs that have DM? So just in the basic way it's supposed to work is your brain sends out signals down the spinal cord, and nerves and muscles send signals back. So there's messages going both directions up and down the spinal cord. And spinal cord is made up of white matter, which is the nerve tracts, and gray matter, which is in the neuron cell bodies. And what we see in the white matter is myelin covering the long threads that make up the nerve cells, which are called axons. And the myelin acts like insulation on an electric wire. So it should look like this, where you've got nice insulation around each one of those wires. A little closer up like that. And so if you have an electric wire that loses its insulation, what happens? It shorts out, and it can't send a signal anymore. So that's basically what's going on in this. So again, this gets referred to in several different ways. Degenerative myelopathy is how it was first described. The chronic radiculomyelopathy was described in 1975, and a German Shepherd dog myelopathy in 1978. It's basically describing the same disease. There doesn't seem to be a sex predilection. The males get it as much as the females. Most of the dogs get it in 8 to 14 years old, 8 to 14 years of age. But the mean age is right around 9 years. And like I said earlier, 6 months to 3 years is the usual survival once the clinical signs start. So this is a cross-section of the spinal cord. This is a normal cord that's age-matched to these not normal cords. And you can see that this is stained blue, and it picks up myelin, and you can see it's fairly similar all the way through. This is a cord that from a dog that has degenerative myelopathy. And you can see there's no blue up here and all around the edges, down here. So all those are places where the myelin is gone. Okay? So any nerve body that's going through that area is shorted out, not working anymore, not sending a signal anywhere. Okay? This is stained in a different way, and it gets dark where things are missing instead of lighter. But again, it's different from what the normal would be. This starts out in the spinal cord, and later there's damage that happens to the nerve and muscle, but that's a later part of the disease progression. Okay. So the initial signs of DM are loss of coordination in the hind limbs. Let's see if I can make this run. There we go. It's a loss of coordination. Oftentimes it's asymmetrical, so one leg will be a little worse off than the other to begin with. It's a weakness, and sometimes you have to look kind of close to see what's going on. There's spasticity. Initially, the spinal reflexes are intact, and one of the key features here is there is no pain in the spine. There's no pain because no message is getting through. Okay? Since that nerve has shorted out, it can't send a signal back that says, ouch. Okay? So nothing is getting through there. One of the things that they were just showing there was that the dog's been dragging its toes, and so it's worn the hair off, and it's got calluses on the foot. And this proprioception deficit is one of the early signs as well. They turn the foot over, and the dog doesn't perceive that its foot's the wrong way around, so it doesn't flip it back. Okay? So there's various stages, and again, this is stage 1 where that we just looked at, where there's some uncoordinated movement, and they can't get all the pieces together. Then stage 2, the hind limbs can't support weight anymore. Sometimes the dogs will do well in a cart like this. They still can use their front end, but you can see there's no feeling there in the back limbs at all. And it's depending really entirely on the front end. All right, in stage 3, it's progressed up the spinal cord, and so the front end is no longer, you see they're supporting the back end entirely. This dog can't stand up at all. And now we're losing the proprioception in the front end. And no feeling at all in the back end. You can see the muscles have also greatly degenerated. There's nothing much left there in the hind limbs because they're just basically flaccid. And there's very little nerve reflex happening there at all. And these are some of the diagnostics that the neurologists do to help them determine exactly what's going on. And then stage 4, they're having difficulty breathing. You can see this dog basically got no muscle tone anywhere, and no pain reflex anywhere. It's just, it's reached a point where it's basically end stage for this. So, like we just showed, we've got an early stage, stage 2, where things are progressing to mild to moderate loss of muscle mass. In the second stage here, sometimes there is urinary and fecal incontinence, sometimes there isn't. Stage 3, we're getting more and more limb paresis. The muscles are all getting flaccid, severe muscle loss, and generally there is urinary and fecal incontinence. And especially with the bigger dogs, people don't go past that. You just can't maintain them. This person did a great deal of nursing with this corgi, and took it late into stage 4, where it's starting to affect the brainstem and all the lower motor neurons, and everything is getting paralyzed. So part of the research was to figure out, does this happen at the same rate in all dogs? Is it different in big dogs or little dogs? And just map out what are the stages and how fast do they go through it. If we're going to create a treatment we have to have some sort of yardstick to measure it by. So Dr. Joan Coates and her associates spent a great deal of research coming up with getting this down to exactly what happens at what stage, from starting with the first clinical signs till the dog is euthanized or dies. And most of them are pretty steady progression. A lot of dogs will kind of maintain a plateau for a month or two or something like that, and then they'll get worse and then sort of plateau for a little while. But the overall progression is fairly steady, down to 10 to sometimes 20 months. So again, it's a multi-system disease. It's starting in the upper motor neurons, in the spinal cord and white matter, and then it moves to the lower motor neurons, the ones that go out to the limbs. We've got axonal degeneration and eventually central brain axonopathy. There are a lot of diseases that actually mimic DM, and that's why it's important to get a good diagnosis. German Shepherds seem to have a lot of different spinal cord issues. So I think a lot of vets and some owners too sort of assume if it's an old dog and it's having trouble in the rear, it must be DM because that's what German Shepherds get. But there are a lot of other issues, and some of these are treatable. So it's important to know what you're dealing with. We can have chronic intervertebral disc disease, a lumbosacral stenosis, spinal cord neoplasia, spinal cysts, bony compression lesions, caudal cervical spinal cord issues, and other orthopedic diseases that can go along at the same time. So what are the diagnostics that can be used? You can do a general radiography, will tell you some issues. Sometimes a CT scan or MRI is helpful. In a few cases, cerebrospinal fluid analysis is useful, and there is also electrodiagnostics that a neurologist can do to see how well is it conducting, how are the nerves conducting a signal from point A to point B. Here's a little bit about some of the other issues. The spondylosis is usually older dogs, and it's usually happening over the vertebral bodies. Sometimes it's an insignificant finding, and it's often associated with an intervertebral disc disease as well. Intervertebral disc disease is a chronic disc degeneration you see in older dogs, mostly in larger breeds, and it also produces a progressive weakness in the rear limbs. So you can see these spaces should be all nice and neat and the same, and this one is pinched out, and so is that one. So if it's pinching the discs, and if you've had a disc problem in your own back, it doesn't feel good. That produces pain, unlike a DM progression is progressive weakness, but without pain. And degenerative lumbosacral stenosis, again, there's pain associated with that. So a neurologist would go through and look at what's likely the base cause there, and rule in things and rule out based on what they see. So our lab at Missouri looked at to see if we could find the mutation that was behind all of this. When we started being able to do some whole genome sequencing and things like that, this is one that people thought would be really difficult to find, and actually it popped up a lot easier than we thought it would. And this is the paper that resulted from it in 2009. There's a single base pair difference in the sequence of this the gene, the SOD1 gene, and that tells us when it changes here, that tells us that it's going to be at risk for developing DM. And initially, I don't remember if I put in the slides for the different breeds. Anyway, so if the dog has a normal copy of the SOD1 gene, it's unlikely to develop clinical DM. Like I said, there are other things that can mimic it, so we have to make sure that we're not looking at something else that looks sort of like DM but isn't really. And it will only pass on a normal allele to its offspring. The dog is carrier, it's far less likely to develop clinical signs, and it can pass on either this bad copy of the gene or the good copy of the gene onto its offspring. And the dog that's at risk has 2 bad copies of the gene. There's no normal instructions telling the body how to behave correctly with respect to what this gene does, and that's where you develop disease. After we've been testing for a while, we found out that there are very few heterozygous or carrier dogs that we have confirmed by histopath that did develop DM, which we didn't really expect. But they develop it much older than the other dogs. They're usually 12 or 14 years old or older, and they develop it, and a lot of dogs don't live that long, okay? So probably if dogs live to be 25 or 30, the carriers would all develop it too. But they don't live long enough to develop it. So, but we have seen it in these dogs, including a few German Shepherds, where we have confirmed it in a carrier. The SOD1 gene is actually the most common mutation that's found by. We now have all these different test companies that are running big panels of mutations, like the Wisdom panel and Bark and those guys. And the SOD1 variant is the #1 disease allele frequency in mixed breed dogs. It's almost 8%, and in purebred dogs, 5.5% of all the dogs they test for anything. So it's widespread. It's not just German Shepherds. It's not a different gene in German Shepherds than it is in everybody else. And this suggests that mutation that's been around for a very, very long time. Before we started sorting things out and said, this is a German Shepherd, and that's a poodle, and that's a Cocker Spaniel, this mutation, it has probably been there forever. And part of that is that there's no selective pressure against it because it's older onset. You don't know that they're affected until they have grandkids on the ground, okay? That makes it really hard to get rid of if you don't have a DNA test. But it also means that, it looks perfectly healthy at the age when you're breeding it. And so even a wild dog or, randomly bred dogs on street dogs, there's nothing there that's going to slow them down from reproducing in the time that they would pass this gene on. And so, it's widespread and it's common. In some breeds, it's altogether too common. And it's in mixed breed dogs. That's one thing that, the people that say, oh, mixed breeds are so much more healthier. Nope, it's there too. So, interpreting these test results, again, testing at risk means a risk for developing clinical signs of DM as they age. And I'm going to talk about how we evaluate that risk in just a minute. The variable age of onset means that not all dogs that test affected or at risk will develop DM because there are some that are going to die of something else before they develop clinical signs of DM. The perfect example is we did a lot of work with the Boxer Club initially on this, and it's very widespread in boxers. When we first started testing, it was probably over 80% allele frequency in boxers. And part of that is they probably had some very prominent, widely used stud dogs that were at risk or affected and later developed it. But they also have a large problem with cancer and heart problems. and those tend to kill them when they're 5 or 6 years old. So they didn't know if those dogs were carrying DM as well. So it actually was kind of a good news, in a backhanded way, that they developed DM because they didn't die from cancer or heart disease, okay? So you kind of have to weigh all these things out of what's going on there. And a dog that dies, that tests at risk and dies without showing clinical signs, it doesn't mean that the test was wrong. It means they just didn't live long enough to develop it. And eliminating all the dogs that test affected or carrier from the gene pool is going to create a horrible bottleneck for the breed, especially, like I said, with the boxers. They need those dogs in the gene pool to breed away from the heart disease and the cancer. And so it's using it wisely and trying to reduce the risk gradually, not taking the sledgehammer approach and say, all right, they're all gone out of the gene pool right now, because that really will damage the overall gene pool. So to try and determine how many of these at-risk dogs really do develop clinical DM in their lifespan, and how much should we worry about this? Dr. Natalie Villani recently did a retrospective study and survey of trying to determine the effect of this mutation on with respect to age, breed, background, and sex. How does that influence the risk of developing clinical disease? And so we looked at dogs that had already been sampled and tested between our laboratory, the testing we do in partnership with the OFA, and tried to make an inclusion criteria that took out the bias. A lot of the samples we get are from veterinarians or neurologists testing because the dog has clinical signs they suspect are DM. And if we just looked at those, it's going to very much skew the study at all, because they already know that the dogs have clinical signs. So we took only dogs that we had sampled. They were tested before they were 6 years of age, when we got the DNA testing done. And then currently we're over 7 years old, preferably older than that, so that they should be showing clinical signs of DM at the time that we were designing this study. And you don't need to know all the details here about how they developed it, whatever. But basically, we had a survey that went out to the dogs that fit this inclusion criteria, asking, did they develop the clinical signs? What clinical signs did they have? What age? What was the cause of death and the age of death? And we had a number of questions so that we could try and tease out what really probably was DM and what wasn't, without doing a necropsy on all of these dogs. And then analyze this with a lot of number crunching. We had the records individually assessed independently by several different researchers, and without knowing what the genetic result was on all of these dogs. So they assigned them as likely DM, possible DM, or unlikely DM without knowing what the genetic results were. Then put the genetic results back together with how they assessed the clinical aspects of it. So we had 325 of the respondents that likely were that did report signs of the clinical myelopathy, and of these, they were determined 115 were likely DM and 210 were unlikely to be DM. And from these, we looked at what was the cumulative risk. So the dogs that tested normal, or GG, you see that line stays pretty much flat. And the dogs that tested carrier, that stays flat until they're about 11 or 12 years old, and then there's a few individuals, but it stays under, I believe they had under 5% that had any recorded clinical signs show up. This little spike here is an artifact of one dog that at age 14 developed some clinical signs at that point. So that's a little artificial. But the dogs that are testing AA or at risk, you can see starting here at 6, they're starting to show clinical signs and or be euthanized because they're showing clinical signs, all the way up to most of the dogs are developing some signs, or over 60% of them. And here's the risk by sex and age. It's kind of the same curve for male and female. There's no statistical difference between the males and the females. I will say we do, when we're doing the testing, we get, each week when we're doing the tests from veterinarians and neurologists that are sent in, it's 2/3 male that we're testing and 1/3 female. And I've been noticing that as I'm sending out reports, and I don't have a good explanation for that. You know, is everybody testing the females when they're young, and you already know what their genotype is, and the males don't get tested till somebody either decides they want to use them for breeding, or they're showing clinical signs later. You guys have a good explanation for that?
Most of us test, if we expect something, we'll test all the puppies like within the first 6 weeks
Yeah.
prior to selling them to somebody.
But that
That
should
way
be
we
equal
can let
male
them
and
know, hey,
female.
this puppy's a clear, or it's an affected,
Yeah.
or whatever.
Yeah, when they're being tested as puppies, that should be pretty much 50-50. So,
Did they make any distinction on the bitches, whether they were in a reproductive cycle or were they stay?
no, we
So there
didn't,
was no distinction?
yeah, no distinction.
Do you think it could be possibly that bitches are protected if they're reproductive phase?
No,
I mean, there are many things that.
no, I'm just looking at what's getting tested. But it's something we've noticed, and I'm not sure that this is, grand research information that we need to know. It just seems really odd that consistently every week we do 2/3 male of all breeds, 2/3 male and 1/3 female. So I suspect that the females are getting tested younger before they're getting bred or chosen to be kept to be bred, but I don't really know. So it's one of those head scratcher things that we're not sure. So the breeds that were represented in the study that Dr. Galani did, the most respondents were from Ridgebacks, Boxers, Bernese Mountain Dogs, German Shepherds, Chesapeake Bay Retrievers, and Pembroke Welsh Corgis.
A friend of mine,
Helen,
very
can't hear
heavily
you.
breeding
Helen.
King Charles Spaniels.
Yeah.
And she said they found that in a tremendous amount of the King Charles Spaniels.
Yeah, she asked about Cavalier King Charles Spaniels. I didn't put a slide in here that I usually have when I'm doing this for all breeds on the breed distribution. The Cavaliers have a huge problem with this. They're right around 80% allele frequency. And I think most veterinarians were taught that this is a German Shepherd problem or it's a big dog problem.
Sure.
And so until we had this DNA test, it wasn't even on the radar screen for most veterinarians for little dogs. But it's actually very common in the Cavaliers. It's in Shih Tzus, it's in pugs pretty commonly. We even had one recently, Yorkie, that was tested as affected and showing clinical signs. So, it really can be anywhere. And that was news to a lot of vets and a lot of breed clubs, I think, too. When I first started talking to the Cavalier people about it, they didn't want to hear about it.
Yeah.
They really didn't want me telling them that something was wrong there. But now they do recognize it, and there are a lot of breeders trying to do some carefully work out of that corner. So when we make a graph here of the disease risk by breed, Looking at the age of onset of clinical signs, this blue line is the Rhodesian Ridgebacks. They tend to show the clinical signs earlier, like right around 7 years, and everybody that tested at risk, all the dogs are basically showing clinical signs and or dead by 8. So it shows up and they're done. The boxers are this one here. There's a little bit more age range from about 7 to 10 when the clinical signs are showing up. And the Pembroke Welsh Corgis have quite a spread, some of them up to 14 years of age before they're showing any clinical signs. Okay? And here's a graph that shows them split out by breed. The red line, here's your German Shepherds. The red line shows all the other breeds, and the blue line here is German Shepherd. So in this study that we did, this retrospective study, most of them were showing clinical signs by around 7 and between 7 and 8 or 9 years of age. Okay? And here's the same evaluation for dogs that are carriers. The few dogs that we had that were determined to be carriers. This is for all breeds. It stays pretty close to 0. And here's 2% here. And Rhodesian Ridgebacks were the one that popped up much earlier on that one as well. And part of the research that we're doing ongoing is there another mutation that's making Ridgebacks more susceptible? The dogs that get it early on, do they have something different than the dogs that are getting it with later onset? So this survey, the basic summary of the results is by 12 years of age, over 50% of the AA dogs develop the DM-like disease, and over 60% of them develop signs when older. Compared to the normal dogs, the dogs that are carriers had an increased risk of developing DM before 12 years of age, but fewer than 5% developed signs of DM by 14 years of age. So there is a slight risk to carriers, but it's very low and late. So we don't recommend taking the carriers out of the gene pool because 14 years is a good long life, and you need some of those dogs. But again, you should be breeding them to dogs that are testing normal, so you're reducing the risk in the next offspring. But don't avoid them entirely. And we didn't find any sex effect on the development of the young. And yeah, again, Ridgebacks had the earliest ages for all the way around. So part of what Dr. Coates has been doing is looking at treatment for this and looking at it as a naturally occurring model for ALS in humans. There are a number of SOD1 mutations that occur in humans, and so this is a naturally occurring model, a spontaneously occurring disease. The rats and mice that they studied, tried to use for models for ALS, they have to create those mice and induce the disease. And most model stuff has really not helped them do anything to help solve ALS in humans or come up with a treatment. So the hope is that by looking at the disease progression in dogs, and since dogs have a comparable size and complexity of the nervous system to humans, we have a large clinical population to evaluate therapies on that, and we live in similar environments since the dogs live with us. We're exposed to the same environmental stresses. If we could develop a treatment that would work on dogs to slow the progression of disease, that hopefully that would work for humans as well. So the working hypothesis was that since it's a naturally occurring animal disease, it will be beneficial in preclinical development of therapies targeting SOD1 to slow the progression of DM and ALS. And so they wanted to evaluate the safety and pharmacokinetics of delivery of targeted SOD1 therapies to come up with a protocol for treatment and demonstrate that there was a therapeutic effect by using these. So the SOD1 mutation is something they call a toxic gain of function, and it's not that we want to stop, it's not that we're losing function, it sort of, you have to think of it sort of backwards. It's gaining function of gathering up these SOD1 globules in the cell. These are normal cells. You see they're completely clean because the SOD1 is getting cleaned out of there. When they have the AA genotype, they're gathering up these SOD1 molecules and it eventually makes the nerve cell die and then it doesn't work. Okay, so they have to look at a way that they can knock down that effect of the SOD1 so it'll slow down the disease progression.
Can you speak up a little louder?
Okay. This is actually Dr. Coates' work, and I'm just sort of using this as an example. What they're trying to do in a treatment is to slow down the disease progression by knocking down SOD1 in these dogs. And so they're targeting exactly what the gene, the mutation in the gene does, and trying to interfere with that. And after doing a clinical trial with, I think they had about 20 dogs in this, they found that the time from onset of clinical signs to not being able to function anymore was a little longer compared to normal controls, but the difference wasn't significant. So they showed that they could treat them safely, but the treatment that they chose didn't actually work the way they were going to. Okay, so that clinical trial is over after Dr. Coates put in a lot of work on that. But they'll use the same delivery method. They're trying to come up with a new clinical trial using a different way of interfering with the what this gene is doing. So again, these dogs that these weren't laboratory dogs; they were privately owned dogs. They continued to show disease progression, but they didn't detect a clinical difference. And you know, if you look at the motor neurons, it was different, but it still wound up with the dogs progressing. So this is not going to work as a reliable biomarker for the disease. But there's more to come. Okay, so how common is DM in the German Shepherds that have been tested? I don't have access to the stats from all the other labs. We're the laboratory that discovered it in Missouri. Then we published the mutation in a scientific journal, and it's not patented in the U.S. because it's considered a discovery, not an invention. And so once it's published, any other lab that can run DNA tests could read that paper and develop a test that they can run as well. So there are a lot of other labs that are running the DNA test for DM. So the stats in our lab, we screened all the German Shepherds that have been submitted for the CHIC program. That's the Canine Health Information Center, which your breed isn't using very much. It would be nice if there were a lot more German Shepherds banked in the CHIC DNA bank. And a lot of breeds do that at their national specialty. They'll do a blood draw or whatever. What the CHIC program is, I'll just add this in here, is it's a DNA bank. You can send any dog in at any time for any reason. And the DNA is banked in one location. Any researcher that has a peer-reviewed funded grant can ask for a portion of that sample. Okay, so one sample doesn't just go to one researcher and sit in their freezer until they retire. It's available to many different researchers for many different projects. And so if somebody wanted to do something on Addison's disease, they'd contact Eddie Zook over at OFA. He'd look through the records and see which fit the criteria for this researcher's project. And then they call our lab and say, we need a portion of the sample from, these 100 dogs to go to this other lab. We had a bunch that went up to a lab that's working on bloat. So he had us get out a portion of, I think it was 120 Great Danes, and those went to another lab. And so it's many different labs and a lot of different uses. I would also say if you are freezing semen on a dog, bank a DNA sample, because 10, 20, 30 years from now, when you still have that frozen semen and there's DNA tests that we don't know about yet, we haven't come up with yet, you can access that DNA to run a DNA test. You don't have to take one of your breeding units and make DNA out of it so you can run a DNA test. So when you're freezing semen, bank a sample, a DNA sample with the Canine Health Information Center. You can do it either by a cheek swab or by a blood sample, but I'll tell you that all the researchers want the blood-based DNA because it will run anything on any platform, whereas the cheek swab won't necessarily work on everything that can be done. So it's not a big deal. Your dogs can handle losing 5 mils of blood, and that will make a lot of DNA that can be used for a lot of different research projects. There's my plug for CHIC. And just go to ofa.org and the DNA bank stuff is there. You can find that. So we only had 317 of your guys in the CHIC DNA bank. 50% of them tested normal, 40% tested carrier, 10% tested at risk. That's probably pretty, most of these samples were taken 10 or 15 years ago. So it might have changed a little bit now, but that's probably a pretty random sample and a fairly good idea of how common it is. And like I was saying earlier, the tests that are coming into us are probably skewed towards the affected dogs because a lot of them are coming in, they're coming into our lab being tested because they're showing clinical signs. So the ones that we get from veterinarians and neurologists and stuff are pretty close to 30% testing effective, okay? In total, we've done pretty close to 25,000 German Shepherd dogs. And that kind of gives you an idea of how common we're seeing it. So it's there, but it's not every German Shepherd. But it's certainly worth testing. Okay, so now what we're doing now so the current grant that is funded by the Canine Health Foundation from the German Shepherd Club of America is identification of genetic risk factors in degenerative myelopathy in German Shepherd dogs. And this is a grant that Kristen Lindblad-Toe from the Broad Institute, Joan Coates from Missouri, and Dr. Gary Johnson at the University of Missouri are working on together. So the aims for this are to perform some health updates for already collected and genotyped German Shepherds and look at the age of onset distribution. And we've done some of that with the retrospective study, but we had really strict criteria there, and that eliminated some of the dogs that we already have sampled. So we're going to take a look at everything that has already been banked to try and include as many dogs as possible. We want to try and identify some additional genes that modify whether the SOD1 mutation leads to DM or not, or affects the age of onset. So are there other mutations that influence the expression of this DM, of the SOD1 gene, that either protect the dogs from showing clinical signs, or is there something that the dogs that get it earlier have a certain mutation, and the dogs that get it later don't have that mutation? You know, there may be additional risk factors out there that are influencing the age of onset. And then to do whole genome sequencing of the German Shepherds that don't have the SOD1 mutation, but do show the histopath signs that say they have DM, and see if there are other mutations that exist in these German Shepherds. We've got two German Shepherd dogs that had classic DM signs, but did not have the SOD1 mutation. They tested normal, and when they looked at the spinal cord section, they had changes that sure looked like it was DM. So what did they really have? And we've whole genome sequenced both of these, and so far we haven't found another mutation that explains why those dogs develop DM. There's a needle in that haystack somewhere, we just haven't found it yet. We also whole genome sequenced a third one that fit that same criteria. It was sent in as German Shepherd dog, I think it was 12 or 13 years old. All the history said that it had that. And we whole genome sequenced it, and nothing looked right on this. And then we tried to align it with the canine reference sequence, and we couldn't even get it to line up with that. And we were really scratching our heads on what's going on here. And we finally, we said, well, could we get a second sample from this dog? Well, no, they euthanized it and didn't save anything. They didn't send anything in for a necropsy, and they didn't save any tissue samples to retest it. So we started thinking, could this be something else? And they did another alignment looking at things, and it lined up 95% match with a horse. So we asked the veterinarian, and they do have an equine practice, but no, they didn't mix up anything. But yeah, they did. So we have a horse that doesn't have SOD1 mutation. That we did a whole lot of work on trying to figure out why it didn't have the SOD1 mutation. Yeah, that was a big surprise, and a lot of money down the tubes, and a lot of head scratching of why did this work. So on this current grant, what we're doing is we have done a pretty good review. We're still looking at more of it, of what has been tested, the samples that we already have for those at risk. And we've got over 5,000 dogs sampled that tested at risk, 5,000 German Shepherds that tested at risk. And so we're looking at which of those dogs are currently over 10 years of age, or I guess actually run down to 8 years of age, and which one of those had clinical symptoms and which didn't. Okay, we've got about 200 of those that we've identified that we have contact information for, and that have not reported. Our records show that they did not report any clinical signs of DM. So a survey is going to go out to those owners of those 200 dogs first and see if, did they develop any clinical signs, or did they stay clinically free of DM until they died, and what age did they die, And that will help us determine what questions we need to ask and how many more dogs that we need to sample to try and sort this out. So the email survey is in preparation. We've had a little difficulty with the IT folks at Missouri helping us with that. But that will go out to all the dogs that fit the inclusion criteria in the survey that we're doing. And then, like I said, we're going to have a more widespread survey based on the initial results from the samples that we already have. If you receive that survey, please, please, please send back the response. What we need from you guys is we need some health updates on previously sampled dogs that had the at-risk genotype. So if you've sent a sample to Missouri or through OFA, had it tested, and it tested at risk, but it was clinically normal at the time you tested it, we need to know about that. And the samples that were, dogs that were tested at other laboratories, so if you tested your dogs through Embark or Wisdom Panel or something like that, we don't have a sample and we don't have access to that sample. If they are older dogs, and especially if they remain normal, well, clinically normal or healthy, we want a sample from those dogs, okay, to include in this research. And then if there are dogs that are 10 years or older and not sampled, we want samples from those dogs. What ideally Dr. Lindblad-Toe wants to do is look at if she can come up with a large number, a matched number of old dogs that have the at-risk profile but do not show clinical signs, then we can compare those dogs to the dogs that developed it when they were 7 years old. Okay, and then that's how we can separate out and look for other mutations that are either making the early ones go early or protecting the ones that are older. She found something like that in Pembroke Welsh Corgis. It makes about a 6-month difference between the age of onset, but it does have an influence on the age of onset and progression of the disease in the Corgis. The problem with boxers and with German Shepherds is we're kind of getting an idea that they aren't, that old age, not affected, but at-risk profile, those dogs don't exist. That's kind of what it's looking like right now. So before we launch full blow into that and say we're going to have that kind of a separation for you, we need to know if those dogs exist. So that's what these initial surveys are for. And we will have a online survey and update thing available, but for now you get to email me and I can answer all your emails. So.
Okay, about 5 years ago or 6 years ago, I can't remember, we had a president of our club that donated through the German Shepherd Dog Club of America quite a large sum of money, about $70,000, down to the veterinarian in South Florida. We never did find out anything what happened with that, if that fellow was still in existence or any of the tests were ever run. I mean, were you familiar with that?
So the guy down in Florida, I believe, has retired at this point. And we offered to collaborate with him and run some of the samples of dogs that he was looking at with the tests that we had, and we'd supply him some samples from the dogs that we've worked with and verified that had DM, and he never took us up on that offer.
Next question. Have any of the results from this DM test been compared to any results from any type of communicable problem in humans? Somebody had mentioned one time to me, and I think it was Lou Gehrig's disease, that they had said there was a lot of similarities between it. Has anybody ever?
So yes, the ALS and some forms of Lou Gehrig's disease are equivalent. That's why Dr. Coates has been working on something that influences how this mutation works in dogs, because it should translate to humans with ALS. There's a lot of spontaneously occurring ALS, or they think it's spontaneous at this point, but there is a fairly significant portion of the ALS cases in humans, that are due to SOD1 mutations. So it is very translatable between. Now in humans, there's many different mutations known in the SOD1 gene that all lead to Lou Gehrig's disease or ALS. In the dogs, we only just knew of one that's widespread in basically every breed. The exception to that is we did find a second mutation that occurs only in Bernese Mountain Dogs, and it's actually quite rare in Bernese Mountain Dogs. The mutation that all the other breeds get is present at about 38% in Bernese Mountain Dogs, and the mutation that only occurs in Bernese Mountain Dogs is under 5% prevalence there. So,
What just seems to me that there are always two separate factions. There's the people that research with the dogs, there's the people that research with humans, and for some reason, they don't get together enough.
yeah, so the dogs and human researchers had been quite separate, but there's more and more move towards this One Health initiative, and actually Missouri has a huge initiative going on called the One Health Initiative, where the human researchers and the animal researchers are working together so that we can look at what overlaps. And dogs are great models. Like I said, we live in the same environment and we're exposed to the same toxins and the same foods and things like that. And so humans and dogs are really going to help humans, and humans can help out the dogs. But some of the things we find in humans, we can take that back and say, oh, is that explaining something we hadn't figured out in dogs? So there's a great deal of back and forth.
Okay, then one other thing, I think we ought to be bringing to the attention of the people who are currently breeding the fact that this is a problem. If you are going to breed, if you have a bitch that you are going to invest the money in breeding, take the time, get the DM test done, and if you are breeding to a male, ask for a DM result. That way you can breed to an effect. I mean, it'd be okay. So if you have a DM clear, you can even breed to an effective if you had to. But you better know what you're doing, because otherwise you're going to lose a lot of puppies.
Yeah, so for encouraging DM testing, we strongly encourage people that are breeding dogs to DNA test and then use that to help make your choices to reduce risk in the upcoming generations. Now, again, we don't want you to use it as a sledgehammer and take everything out at once.
No.
But use this wisely. And also sometimes it gets too far the other way, and people are breeding a test result instead of breeding a dog.
Yeah.
This is one piece of the whole puzzle, and you have to breed a whole dog. You can't just breed a test result and have any
But the
success.
big problem is
So-
people who don't test, if they have an affected female and an affected male.
Right.
And these people sell puppies to people, and those
So-
poor dogs
So the thing
can't
that,
walk.
yeah, the thing that really needs to happen is the public needs to be aware to ask for results.
And ask for DM clearance on
Right.
puppies if they're buying.
So-
Yeah.
You had a,
Yeah, I'm just curious, like, a lot of people said that you are more distant on the dogs and on the humans. You know, are people testing the dogs more than they're testing their bitches?
So, the samples that we're getting from veterinarians and neurologists, where the dogs are showing clinical signs and they're running a test to see is it actually DM or is it something else, those have been 2/3 male and 1/3 female. And that just seems weird to me. The ones that we're testing through OFA, I just brought this up in the last few days. I don't have the stats from Eddie Zook to know if the ones being tested through OFA are 50-50 or more females or something that are being tested. Because since they can do it with a cheek swab, I think a lot of the young dogs are tested through OFA. And so it could be that that's where all the females are getting tested. I just don't know at this point. But when we looked at whether the males show clinical signs more than females, no, there's no difference. They show the clinical signs, same age and same percentage.
I missed part of the discussion. One of the important parts for me and maybe you can explain it to everybody else. This testing that we're doing now is the same testing or similar testing that was done on corgis. And if you could explain that which they found in the corgis and how it would be similar to what we're looking for here.
Okay, so what Dr. Lindblad did with the Pembroke Welsh corgis is there was in the Pembrokes there is a large range of age of onset, and so she took a group of the dogs, all of them testing at risk. A group of the Pembroke Welsh corgis that showed clinical signs when they were 7 or 8 years old, and another group of genetically at-risk Pembroke Welsh corgis that didn't show any clinical signs till they were 13 or 14 years old, or were 14 years old and still weren't showing any clinical signs. And she compared those two groups to look and see if she could find a mutation, and she did find another gene that seems to influence the age of onset. So the dogs that are getting it much later seem to be protected if they have this gene, this mutation. Whereas the dogs that are getting it earlier influenced by this secondary gene. They all have the SOD1 mutation, but some of them have this secondary gene and some of them do not. So that's what we're trying to mimic here. But the first thing we have to figure out is, do you have that old group that was really easy to find in Pembroke Welsh corgis? We wanted to do the same thing in boxers. The old group doesn't exist, so we weren't able to do that. And so, does that exist in German Shepherds or not? That's our first step here, is to figure out, do we have enough old dogs that we can make a comparison between those two groups? Anybody else? Here.
Yeah. What's the SOD1B gene?
So she asked what the SOD1B gene is. That's the one that's found in Bernese Mountain Dogs. Okay? So we had a Bernese Mountain Dog that had classic DM, didn't have anything else that looked like it could be influencing that. When we looked at the spinal cord after that dog died, it had classic histopath that said, yep, this is DM. So we resequenced the SOD1 gene, and we found a second mutation on the same gene. Okay? And it also means that there's no correct instructions, and so things go haywire, and those Bernese Mountain Dogs show that. We tested 1,000 Greater Swiss Mountain Dogs to see if it was there. Every one of them was normal. And so we haven't, and we've tested a number of dogs that showed what looks like DM, but tested normal for the common mutation. We've tested a lot of dogs from other breeds. Nobody else seems to have that mutation. It's only Bernese Mountain Dogs. We've never seen it in anything but a Bernese Mountain Dog. So there could be other cases like that where there is a rare but yet undiscovered mutation somewhere else in the SOD1 gene that's causing that DM. We don't have evidence for that yet. We keep looking for it, but we don't have evidence for that yet. The other thing we found out with the Bernese Mountain Dogs is that if they're a carrier of the common mutation and a carrier of the SOD1B mutation, those dogs also have no normal instructions, and they also get DM. So we call it a compound heterozygote. They also get the disease. So.
Has any testing been done on looking at sibling groups who have tested AA?
She asked if any sibling groups that all tested AA, if we've looked at what specifically?
If you're looking at age of onset or...
Age of onset, yeah.
I have noticed in several dogs that I have tested, age of onset was different for several siblings in different heritage. They tested AA and pitch.
So, let me just interrupt you there.
Showing something aesthetic, the next pitch, our sibling.
Oh, well, you need to send me an email. Yeah, no, we haven't looked at that because generally we don't get that kind of information. So if you have sibling groups with all testing AA and having different ages of onset, we want to hear from you. So write that email down and please send me an email. Yeah.
Those are things that still need looking at.
Yes, absolutely, and those would be a really good group to look at.
Family groups like mother, daughter.
If we can get DNA on all those dogs, it would be good to have. Yeah, we've never turned down a DNA sample. Dr. Johnson's never met a DNA sample he didn't like. So, anything can be useful eventually. All right, so I think that probably,
Somewhere, what, can you look at it? If you can find it, locate it, and then send it to you?
yeah, just send me an email and we'll see what we can figure out. All right, well, thank you for your attention, and hopefully we'll figure out more things for you, and I can go back and tell you we solved everything.
Thank you very much.
Sure.